Going back about a half dozen videos now, I was talking about a structure called the cytoskeleton. And I said the cytoskeleton does for the cell what our skeleton does for us. Our skeleton gives us structure and it allows us to move. The cytoskeleton does the same for the cell. It gives it structure and it allows it, in some circumstances, to be able to move around. Now, on that cartoony diagram that we started out with, remember that idealized animal cell, ceci n'est pas une cellule, right at the bottom? You could only see a few individual little strands of protein, a few microtubules in the background showing you the cytoskeleton. It gives you the impression it's like, okay, a strand here, strand there, strand here, that's the cytoskeleton. But here on this diagram, you can see cytoskeleton fibers lighting up with fluorescent microscopy. And you can see that all of these glowing strands, these are the fibers of the cytoskeleton. And there are a lot of fibers. There are fibers going every single which way. Is a giant spider's nest of fibers inside the cell. So there is a lot going on with this cytoskeleton. It is a network of protein fibers extending throughout the cytoplasm. It provides mechanical support for the cell. Part of the reason for that is because it is made up of those nice sturdy microtubules with that nice cylindrical structure to them. If I were to take a sheet of paper and I were to try to get it to support some weight, it doesn't have enough structure to do that. It's a little bit flimsy for that. But if I were to take that sheet of paper and I were to roll it into a cylinder, well, now it's got a little bit more sturdiness to it. It can resist. It has compressional support. I could probably even put a book on top of that sheet of paper, you see? So arranging a protein into this nice kind of cylindrical column, like a microtubule is, gives you that nice compressional support. So it provides mechanical support for the cell. You have smaller fibers, which we call microfilaments and intermediate filaments, which provide tensional support. They prevent the cell from being pulled apart. It helps maintain the cell's shape. It also keeps all of the organelles in the correct position. It kind of anchors them in place. And it is dynamic. Dynamic means it's able to move, so it allows the cell to change its position. Changes in the cytoskeleton contribute to the motion of amoeba, what we call amoeboid motion. If you've ever seen a video of an amoeba moving around, it's really cool. They send a pseudopodia out in some direction and they kind of pull the rest of their body up to meet that pseudopodia, and then they send up another arbitrary part of their body and then they pull the rest of themselves forward, and they're this kind of gelatinous goop sort of crawling their way haphazardly through their environment. And the way that they're doing that is by manipulating the proteins in their cytoskeleton. And a lot of those proteins are the same proteins that you use inside of your muscle cells to be able to move around. So amoeba, even though they look like they're moving around, shambling around very, very strangely, they're actually doing something pretty darn similar to what you're doing when you walk around. So I mentioned that there are a couple of different kinds of proteins giving you two different kinds of support. We have tensional support and compressional support, and I like to use the analogy of a suspension bridge here in order to talk about these two different kinds of support. So microtubules are straight and hollow. They're made from a protein called tubulin, and they can help guide the movement of organelles and chromosomes. Later we're going to talk about mitosis in this unit. It's coming up pretty soon, actually. And is microtubules that help guide chromosomes and separate chromosomes during that process. So they are big girders. On a suspension bridge, they'd be these big pillars, the green pillars here on this diagram, that hold up the suspension bridge, right? What's the force on this pillar, right? The suspension bridge is pulling down on it, so you have force pushing down, and then you have the force of the ground pushing up on there. So it's being compressed. The forces at play are trying to crush that green pillar, but it's sturdy enough to be able to resist that. Now, the suspension parts, the red filaments, the wires, are having the opposite problem. They have the weight of the bridge pulling down on them, and then they have tension from the cables up above pulling up on them. So they're not being compressed. They're actually being pulled apart. They're being pulled away at both ends. They need to offer tensional support, and that's what the intermediate filaments and microfilaments of the cytoskeleton do. These are smaller proteins than the microtubules, but nonetheless very, very important. They are made from a fiber called actin, and they are thinner and they add tensional support and allow for contraction. Actin is one of the filaments, one of the proteins that is involved in your muscle cells that allows you to move, and this is it over here on the left-hand side. So actin can grab onto other proteins. This is myosin that's grabbing onto, and it has these little heads, and these heads contort down. They kind of pull themselves along that other protein, and when they do that, they're able to contract. They're able to move themselves along, and by contracting themselves or moving themselves across this protein, you're able to change the shape of how the cell is situated. You're able to shrink the length of your muscle cells, and that allows you to, say, lift up something heavy with your arm by contracting your bicep. And this also allows cells to be able to move themselves around as well. Here in the upper right hand corner, I have a happy looking little protein. It's got a little strut going on. What is it doing? This is a kinesin protein, and it is walking on a microtubule. So it's walking on the microtubule like it's a little pathway there, and it's dragging a vesicle along with it. So this is another service that the cytoskeleton can actually provide. It becomes a kind of monorail system around the cell. The cell can use this as a railway, a train system, to drag vesicles from point A to point B. Remember how the Golgi apparatus sends all of these vesicles, all of these chemical products of the cell to their final destination? Well, it needs a way to do that, and the way it does that is by sending vesicles along these microtubule proteins. But remember from that slide with the fluorescent micrographs, you could see that the fibers went absolutely every which way all over the place? Well, that is a really convenient network for these little proteins, these little motorized proteins, to walk along and drag these vesicles absolutely everywhere where they want to go. So it's a transport network as well as being a support network and being a method of movement for the cell. Okay, we got 3 different kinds of proteins that are involved in this cytoskeleton. We talked about microtubules, microfilaments, and intermediate filaments. The microtubules are made out of a kind of polymer which we call tubulin. This is a protein called tubulin. They are structurally hollow tubes, cylinders, and their wall consists of 13 columns of tubulin molecules. So there's a lot of polypeptides in that structure. Here it's talking about the protein subunits. That's more detail than I would ever expect. And it talks about the main function, compressional support, cell motility, as in cilia or flagella. I'll talk about cilia and flagella in a second, but you can see that microtubules are involved there as well. Chromosome movement and cell division. Again, we'll get to cell division in a second, but I already told you that they help separate chromosomes during cell division. And organelle movement. They help anchor the organelles in place, but if an organelle needs to move out of the way, like in cell division, they are going to be the structures that help out. So you can see here that microtubules, they're kind of a big deal. Of all of these filaments, I would want you to know about microtubules the most. Microfilaments, actin filaments, these actin is a really important protein when you get to anatomy and physiology. It contains 2 intertwined strands of actin, each a polymer of actin subunits. The protein is actin, naturally enough. It helps maintain the cell shape, tension-bearing elements. It changes the cell shape, so this is part of the reason why the cytoskeleton is dynamic. It aids in muscle contraction. It aids in cell motility, like when it extends the pseudopodia. Remember when we were talking about phagocytosis and it extended those pseudopodia out to grab some food and then it made that vesicle to pull it all in? Well, the way it was changing its shape to grab it was with these actin filaments, with these microfilaments. This is how the cytoskeleton manages to change its shape and helps in cell division in forming the cleavage furrow. We haven't gotten to that yet. The cleavage furrow is right where it's actually going to pinch itself into 2 different cells, so it's how it kind of cuts itself in half. And something called cytoplasmic streaming, which has to do with the flow of the cytoplasm throughout the cell. Intermediate filaments are fibrous proteins supercoiled into thicker cables. They're thicker than the microfilaments are, makes sense, microfilament, intermediate filament. They are going to be made up of a couple of different proteins, perhaps keratin, and keratin is the protein that makes your hair and nails, fingernails, nice and hard. It also plays a role in keeping your skin nice and waterproof. These intermediate filaments play a role in maintenance of cell shape, anchorage of the nucleus and certain other organelles. So again, the cytoskeleton anchors your organelles in place, and the formation of the nuclear lamina. What the heck is that? The nuclear lamina is this meshwork of fibers that's inside the nucleus that helps give it some additional support. You guys don't really need to know about it. So these fibers are involved in cellular motion in a couple of different ways. We talked about movement around the cell, and we talked a little bit about amoeboid motion. So here is a video of an amoeba moving around, and I decided to get a little bit poetic here. So aggressive amoeba amble along apace. And you can see here that this amoeba is using the dynamic properties of the cytoskeleton to be able to contort its body and move itself around its environment. But you can use the microtubules in a different way. You can use them as cilia or flagella to propel yourself around if you're in an aquatic environment. So cilia and flagella are kind of external features of a cell. Usually you find them on single-celled organisms, but there are some exceptions, like we have cilia on some of the cells in our trachea. But these are two methods of propulsion. So we have aggressive amoeba amble along apace. Alternatively, playful paramecia prefer to propel post-haste. And paramecia have cilia. Cilia are small hair-like structures that cover the outside of their cell bodies, and they move back and forth in a coordinated type motion to allow them to kind of swim around. In the description, you will find a link to a video from Jam's Germs. This is a channel run by a microscopist named James Weiss, who handles all of the video footage for a channel called Journey to the Microcosmos. You might know that one better. Both of them are really, really cool channels. In this particular video, he managed to capture the rhythmic motion of cilia on a Blepharisma protist swimming around in the water just really exceptionally well. You can clearly see the cilia going around in this wave-like motion as it swims around its habitat. Very cool stuff. So cilia are one way that you can move around your environment, and another one would be flagella. So flagellum, again, this is some Latin roots here. To flagellate is to whip, so a flagellum is a whip-like structure. It's not a whole bunch of these tiny little hairs. It's a single long whip-like tail at the end of a single-celled organism. And it's used to go back and forth and back and forth and back and forth, and that single large structure whipping back and forth like that is sufficient to propel it forward with great speed, actually, quite frequently. And cilia and flagella have the same basic architecture. They're made in basically the same construction. It's just that flagella are much, much longer, and cilia are shorter but more numerous. Cilia may extend out of non-moving cells as well. Flagella you're never going to see them from non-moving cells, but cilia, you'll see them, like I said, coming from your trachea and things like that, because sometimes you need cells that can sweep materials past them, and cilia might be really, really handy for that sort of thing. Here we have a human sperm with its flagellum. This is the only cell in the human body that would have a flagellum. So if you think back to that idealized animal cell, right, and one of the structures that it had was a flagella, well, that's because you would only ever see a flagellum on an animal cell, never on a plant cell. But that does not mean that all animal cells have flagellum. In humans' case, only sperm cells would have a flagella. So these pink colored cilia down here, these are the ones I was talking about on the respiratory tract. So if you inhale a little particle of dust or something, it's going to get caught in the mucus in the lining of your respiratory tract, and that's going to catch it so it doesn't damage the alveoli in your lungs, and that's great. But then you have this little wad of mucus there. So what do you got to do? Well, these cilia can help bring that up. They're going to move in that kind of rhythmic wave-like motion in order to start bringing it up, and then you can start coughing and expectorating constricting, expectorating, coughing it out of your system. So you can cough up a little bit of mucus there, nice and handy. And then this, what looks like a very hairy cucumber here, is a kind of protist, and this is the cilia on the protist. And the reason why it looks like this is that this is a scanning electron microscope, and this is what that organism looks like in 3D, not in cross-section. Whenever we look at something under a light microscope, we're essentially seeing a cross-section of it, and that's true when we look at it under a transmission electron microscope as well. But this is scanning electron. You can see here it says colorized SEM. So we're going to get a 3D surface image of what that thing looks like, and you can see that the cilia actually have a little bit of thickness to them there as well. Now you may remember that prokaryotic cells had flagella also. So you might wonder, is it the same as the eukaryotic flagella? And no, it is not. It's not the same. Prokaryotic flagella have a single protein filament called flagellin. The protein is called flagellin, and it can spin 360 degrees. So it's anchored into their cell wall, and it has a motor protein that's attached to it, and it literally spins around like a little propeller, right? So like a speedboat propeller, just spins around and propels that bacterial cell or archaea cell forward in the water. But that system would not work for a eukaryotic cell because this is a very, very small flagellum, and eukaryotic cells are much, much, much bigger than prokaryotic cells are, so that wouldn't be able to propel it. And if we tried to scale it up, the swinging this giant flagella around like that on a motor protein, the shear forces would just cause it to fly right off. The thing would just tear free from the cell. Structurally, it just wouldn't be a very sound design for something as big as a eukaryotic cell. So instead, eukaryotic flagella are larger, and they're made of, you guessed it, microtubules. Very important protein, the microtubules. They're made of microtubules in what we call a 9-to-2 arrangement. There are 9 pairs of microtubules going around the outside of this flagellum here in the structure. You can see it here on the diagram. And then there's this central pair of microtubules here at the center. And this makes it sturdy enough, right? This gives it enough structural support to be this giant structure, this big tail hanging off the end of the eukaryotic cell. And then essentially, we just need to wag this tail back and forth. We're not going to spin it like the prokaryotic flagella. We just need to wag it, shift it up, shift it down, shift it up, shift it down on either side so that it can whip back and whip forth and whip back and forth and whip back and forth and back and forth and back and forth. And in that type of motion, we are able to get forward propulsion rather than spinning around 365 degrees. So yes, prokaryotic cells and eukaryotic cells both can have flagella, but there are some differences. Prokaryotic cells almost always have more than one flagella when they have one. Animal cells typically are going to have one. Protists can have more than one. Prokaryotic flagella spin 360 degrees. Eukaryotic flagella whip back and forth. Prokaryotic flagella are made out of a single protein filament. Eukaryotic flagella are made out of microtubules in a 9-to-2 arrangement. So big differences between the two structures.